I would like to introduce our presenters today, Josh and Danya. Josh and Danya are PhD candidates in the lab of Dr. Martin Stampley in the McMaster Immunology Research Center. They are both head teaching assistants for HealthSci 3IO3, McMaster's flagship immunology course. Josh's thesis focuses on understanding the effects of cigarette smoke on antiviral host defense in the upper respiratory tract, while Danya studies the chronic development of smoking-associated cardiovascular diseases such as atherosclerosis and abdominal aortic aneurysm. Please welcome Josh and Danya.
Thank you very much, guys. Thanks for that lovely introduction. So my name is Josh, and today I'm going to talk to you a little bit about the applications of antibodies in health and research, and I'm going to specifically talk about their role in research and diagnostics. So Dr. Ask said that everybody likes training presentations, so just as a brief background, my PhD thesis focuses on investigating the effects of cigarette smoke in the upper airways and their consequences to antimicrobial host defense. And I do this with Dr. Martin Stampley up in the McMaster Immunology Research Center, just on the 4th floor above us here. In addition, we've started a really exciting collaboration with Dr. Ask and Olivia, in which we'll be investigating the mechanisms by which cigarette smoke exposure can predispose to idiopathic pulmonary fibrosis. So we're really excited about that. And as was mentioned, Danya and I are both head TAs for Health Sciences 3IO3, Introductory Immunology. If you haven't taken the course and you have any interest in immunology, I highly recommend it. It covers the fundamentals of the immune system, how it's relevant in our daily lives, and the mechanisms by which the immune system functions. So I would highly recommend taking it if you get the chance. Okay, so in medically related fields, there is a variety of instances where we may have to detect proteins in a complex solution. For instance, if we want to know whether or not there's allergen contamination in our food, right? Individuals that are peanut allergic can't consume peanut antigens or peanut proteins at risk of death via anaphylaxis, right? So we need to be able to understand whether there's peanut antigen in our food, but we can't see that peanut antigen, right? And so we need a biochemical assay in order to detect that. Can we determine if an individual is pregnant before they begin to show outward signs? There are biomarkers, human chorionic gonadotropin in the urine, which can be used to detect pregnancy before outward signs begin. But again, we can't see that within the urine. We need to be able to analyze it. And again, we need a biochemical assay to do that. Can we determine an individual or whether or not an individual who is HIV infected has progressed to AIDS? And this involves the depletion of a specific immune cell subset known as CD4 T cells. Again, we can't see the CD4 T cells in the blood. We need a way to analyze them. And so it turns out that this molecule, the antibody, can be used to detect molecules of interest and to answer these questions. So before we get into how the antibody can be used to answer the question, we have to establish some basic principles about antibody biology. So what is an antibody? So the immune system can be broadly broken up into two components. We have the innate immune system and the adaptive immune system, and they do work together, but broadly they can be classified into two aspects. So the innate immune system gets activated within minutes to hours following exposure to a pathogen, for instance, and features the activity of cells such as macrophages, neutrophils, and alike. Adaptive immunity occurs in a delayed fashion. So 5 to 7 days after you get exposed to a pathogen, you'll have the activation of two major arms of adaptive immunity, T cell responses and B cell responses. And the B cell response is the source of antibodies, so that's what we'll focus on. So throughout our bodies, we have naive B cells, and on the surface of these naive B cells, there is the B cell receptor, okay? And this is the membrane-bound form of the immunoglobulin, or sorry, the antibody. And each of these B cell receptors has two target binding sites, and these receptors are generated randomly during B cell development. So they may be specific for any protein, polysaccharide, nucleic acid under the sun, but before they're activated, they're not secreting antibodies. If, for instance, a bacterial pathogen comes along and initiates a sequence of events that triggers B cell activation, that naive B cell, if it's specific for that bacteria, will become activated and transition into a plasma cell. it will lose expression of BCRs or B cell receptors on the surface, but begin to secrete large amounts of this new form, antibody. And the antibody and the B cell receptor will have the same specificity. They'll both be specific for the pathogen, right? But now we have it produced en masse in a secreted form. And so this antibody, if it interacts with the pathogens, can have a variety of effects, including inhibition of colonization or infection of mucosal surfaces, and it can also promote pathogen clearance by immune cells. So interestingly, antibodies have a very narrow specificity. So on the left-hand side, let's say we had an antibody response induced by Streptococcus pneumoniae, which is a common pneumonia-causing bacterial pathogen, Serovar 6a, so it's a subtype of strep pneumo. The antibodies that are produced will be highly specific to that serovar and will very likely not be able to bind even highly related serovars, such as Serovar 6b. So even though this is the same species of bacteria, and even highly related within all of the different subtypes of bacteria, those antibodies, they have a very narrow specificity and they won't be able to cross-react in general. There are exceptions to that rule, but in general, the specificity of antibodies is quite narrow. And this can be used to identify molecular targets in additional applications, such as research and in diagnostics, with confidence. So before we move any further, there's a few terminology bits that we'd like to go over. Antigen. So an antigen is any macromolecule against which an antibody response can be generated. And you can see the colors there indicate where the word antigen comes from, antibody generator. This can be any protein, nucleic acid, lipid, polysaccharide, etc. An epitope is a region of an intact antigen to which an antibody can bind. Okay, so an antigen may have hundreds or thousands of epitopes. They're just portions of an antigen to which an antibody can bind. Okay, the next distinction is a monoclonal versus a polyclonal antibody solution. So what I've demonstrated here is a model antigen, tumor necrosis factor alpha. And this is a pro-inflammatory cytokine that's very important in sepsis and rheumatoid arthritis, but for here it's just an example. And on TNF-alpha, I've illustrated 4 epitopes in different colors. So in a monoclonal antibody solution, we have an antibody, or sorry, we have a mixture of antibodies, but they're all of the same specificity. So they can only bind to that one epitope, in orange, of that one antigen. They can't bind to additional epitopes in the different colors. They can't bind to additional epitopes on the same antigen or on different antigens. They can only bind to that one epitope of that one antigen. In polyclonal antibody solutions, we have a mixture of antibodies of different specificities, right? So we could have a mixture of antibodies that could bind to all of the different epitopes on this model antigen, or we could have a mixture that could bind to a variety of epitopes on a variety of antigens. However, for the purposes of research, diagnostics, and treatment therapies, as Danya will talk about, monoclonal antibody solutions are most frequently used for their consistency. So that's what we'll be talking about for the rest of the day. So now we can kind of update our statement to say the narrow specificity of antibodies can be used to detect antigens of interest. Okay, so in the research setting, as often happens, my supervisor, Dr. Stanfley, may come up to me and say, Josh, I need you to quantify the amount of TNF-alpha in these new serum samples as soon as possible. And I'll say, absolutely, Dr. Stanfley, let's do it. And to do this, we will use an enzyme-linked immunosorbent assay, or an ELISA. And we'll perform this assay in a 96-well plate, as is indicated on the right-hand side. And so what I'm going to show you is one well of that 96-well plate, and we're going to be demonstrating a sandwich ELISA. So the first step is that we take monoclonal antibodies specific for TNF-alpha, and they're adsorbed or adhered to the solid phase, right, the bottom of the plate. The plate is charged in a manner that the first thing that gets applied to the plate will stick independent of what that thing is. So those monoclonal antibodies are adhered to the solid phase. Next thing that we do is we add our sample on. So in this case, it's serum. And we want to measure the amount of TNF-alpha in the serum. And so the idea here is that the monoclonal antibodies will only bind the TNF-alpha among all of the other components of the serum. So that when we wash away the rest of the serum, that TNF-alpha will remain bound. And you can see here immunosorbent. We're using an immunological reagent up in the title there to adsorb TNF-alpha to the bottom of the plate. The next thing that we do is we add a second monoclonal antibody specific for TNF-alpha. And this one is conjugated to an enzyme known as horseradish peroxidase. There are other enzymes that will do the same job, but this serves as a colorimetric enzyme that will be used in the next step. And so what this does is it establishes a correlation between the amount of TNF-alpha in the plate with the amount of colorimetric enzyme in the plate. So that's the first step of the correlation. So the last step of the ELISA is that we add a substrate that's normally clear but becomes cleaved by horseradish peroxidase in order to produce a blue color. And so now we've extended the correlation between TNF-alpha and the horseradish peroxidase to the amount of blue signal that is produced within each well. So if we were to look at a 96-well plate, this is how we would analyze the serum that Dr. Stampley asked us to. And there's a few different components here. The first is the standards. So on the left-hand side, what we do is we take a known amount of TNF-alpha and we serially dilute it within our wells, and then we run our reaction. And so what we can do is establish a correlation between the amount of TNF-alpha, a known concentration, and the optical density that we get when we read our plate with a spectrophotometer. So on the right-hand side there, you can see a standard curve or a reference curve, and we can use that curve as a comparator. So the next thing we do is we take all of our serum conditions, right? We take the optical density from each of those wells, and we use that optical density to reverse calculate a concentration. And so from this, we can see serum condition 1 is a darker blue, therefore it'll have a higher optical density, and therefore it will have a higher concentration of TNF-alpha than serum condition 1, which is a lighter blue color. And so from that, we can calculate how much TNF-alpha is within each sample. So that's how we can use ELISAs in a research setting, right? In terms of diagnostics, there are a variety of diagnostic tests that rely on this principle. If we wanted to detect malaria, whether an individual has malaria, we can use ELISAs to look for antigen from Plasmodium, the parasite that causes malaria, in the bloodstream. Similarly, we can use antibodies to detect other antibodies to diagnose a lot of diseases. So instead of directly looking for antigen from the pathogen, we're looking for antibodies that are produced by the body's response to the pathogen. So for instance, if we find dengue virus-specific antibodies using ELISA, we would assume that that individual has been exposed to dengue virus. Similar for Mycobacterium tuberculosis. And for rheumatoid arthritis, we're looking for autoantibodies. So again, the ELISA can be used to diagnose a variety of conditions. And in terms of food production, it can be used to detect whether or not there's allergen contaminants within certain food products, again, as we mentioned before. And surprisingly, ELISAs can be used at home quite frequently. They can be bought at Shoppers Drug Mart, for instance, in the form of pregnancy tests. And so if you were to go and purchase a pregnancy test, this is what it would look like. It would have a case, there would be a sample pad exposed, and there would be 2 bands visible, bands 1 and 2. And the instructions would say that if no pregnancy was present, only the further band would turn color. And if pregnancy was present, then you would have both bands turn color. And so this isn't magic. It's a lateral flow ELISA for the detection of human chorionic gonadotropin. And this is produced by placental cells and is a specific biomarker for pregnancy that's detectable in the urine. Okay, so if we took the case off, what we would find is a few more components. We have the sample pad where urine is applied again. We have a conjugation pad, and so this contains anti-HCG monoclonal antibodies that are conjugated to a colorimetric enzyme. So this is very similar to our detection antibody in our ELISA. And it's important to note here that this is an IgG type antibody. It's known as the isotype of the antibody, and that'll become important in a second. At band number 1, we have a second anti-HCG monoclonal antibody, but this time it's adhered to the band. At band number 2, we have an anti-IgG monoclonal antibody. So again, note that our detection enzyme is an IgG monoclonal antibody. And finally, we have an absorbent pad, and this serves to wick the urine along the test. Okay, so what happens? If pregnancy is present, urine is applied to the sample pad, and HCG will be present. And the liquid aspect of the urine can solubilize the detection antibody at the conjugation pad, and that detection antibody will bind to its cognate epitope on the surface of HCG. So that's step one. Step two is this immune complex will move to band number 1. And at band number 1, we have a second monoclonal antibody against HCG. And this will bind to a different epitope of HCG. They're not going to compete with each other because, again, they're different specificities, even though they bind the same antigen. And so now we have essentially our sandwich ELISA format being created. All we are missing is that substrate changing color, which we will now have. So we have a substrate that's embedded in band 1, and the enzyme that's associated with the detection antibody will facilitate a color change reaction at that band. In addition, we have an excess of this detection enzyme or detection antibody that's always placed within the test. And so that will move past band number 1 and become bound by the anti-IgG antibody at band number 2. And this serves as a positive control for the test, right? If the detection antibody can be solubilized, can move past band number 1 where we have our investigation of whether or not HCG is present, to band number 2, then we should be able to interpret the results at band number 1. So we can only interpret the results of band number 1 if band number 2 changes color as well. So if no pregnancy is present, we don't have HCG in the urine, we'll still have the liquid aspect of the urine solubilize the detection antibody and carry it past band 1 to band 2. Band 2 will change color, but band 1 won't change color because you don't have the HCG to link the two HCG-specific antibodies together, and therefore associate the colorimetric enzyme with band number 1. And so, again, if no pregnancy is present, you only have the further band, the positive control band, turn color. But if pregnancy is present, you'll have both bands turn color. The first band, because HCG can link the detection antibody to the substrate at band number 1. Okay, so we've talked about how we can detect proteins in complex solutions, such as TNF-alpha in serum, such as allergens in food. We talked about how we can determine if an individual is pregnant. To answer our third question, can we determine whether or not an individual has acquired immunodeficiency syndrome? There's no soluble biomarker for AIDS, so this presents a problem. We can't just use an ELISA to answer this question. So human immunodeficiency virus, HIV, infects a cell known as a CD4 T cell. And in newly infected individuals or healthy individuals, the count of CD4 T cells is between 500 and 1,400 cells per millimeter cubed of blood. Over time, if untreated especially, the HIV can infect the CD4 T cell repertoire and begin killing those CD4 T cells. And if this happens for long enough, we can get the depletion of the CD4 T cell repertoire to a point where the individual is severely immunocompromised and is predisposed to diseases such as pneumonia. And this is what's termed acquired immunodeficiency syndrome. So what we want to detect to determine whether or not somebody has AIDS is how many CD4 T cells are there? So how can we count CD4 T cells specifically? And to do this, we can use a really fascinating technique called flow cytometry. And this involves using antibodies to identify antigens on the cell surface and thus to identify specific cell types. Okay, so one drop of blood, regardless of where it comes from, will include a diversity of cells. We've got red blood cells, we have innate immune cells, we have adaptive immune cells of every type and flavor. But we just want to know how many CD4 T cells are there. So if we take a closer look at the surface of CD4 T cells, we can see that there are molecules of interest here. We have the T cell receptor. We have the CD3 molecule, which is an important signaling component of the T cell receptor complex. We also have CD4, which is a co-receptor for the T cell receptor engaging with other cells. And it turns out that CD3 and CD4 co-expression uniquely identifies a CD4 T cell. So how can we use that to our advantage? Well, what we can do is we can take this complex mixture of cells and to that mixture add 2 antibodies. In reality, there would very likely be more, but at its most basic form, we could add 2 antibodies. So what we would do is we would add a monoclonal antibody specific for the CD3 protein. And it would be conjugated to a fluorescent molecule or fluorophore. And in this instance, it would be green. Could be any color, but here it's green. In addition, we'll add an anti-CD4 monoclonal antibody that's conjugated to a different color fluorophore. So now what we've done is associated the color green with the ability to bind CD3, and we've associated the color red with the ability to bind CD4. And so when we put this in the mixture, there's three different scenarios that may happen. On the left-hand side, we can see cells that do not express either CD3 or CD4, and no antibodies will be bound to the surface of those cells. On the right-hand side, we can see our CD4 T cells that express both CD3 and CD4, and we can see that both the green fluorescent and the red fluorescent conjugated antibodies will bind to that surface. And in addition, to provide a little bit of context for the next slide, there are other types of T cells which express CD3 but not CD4, and so in that case we will only have the green fluorescent antibody binding to the surface of those cells, but not the CD4 binding red fluorescent antibody. So now we take this mixture. Some cells will be stained with antibodies, some cells will not be stained with antibodies, and we analyze them one cell at a time. We pass them through lasers, which excite those fluorophores and cause those fluorophores to emit light of a specific wavelength. And using a flow cytometer, we calculate how many cells fluoresce both red and green, indicating CD3 and CD4 co-expression. And so this is where the term flow cytometry, cyto referring to the analysis of cells, and flow refers to the manner in which they are analyzed. They're sent through in a liquid stream through a series of lasers in order to analyze what antibodies are bound to the cell surface. And so this is what a flow cytometry plot might look like. We have two axes, and on the bottom we have the green fluorescence intensity, and on the y-axis we have the red fluorescence intensity. And so the concept here is that cells and each of those dots is a cell. So cells that express CD3 will move to the right, okay? So on the bottom right-hand side, we have cells that are expressing CD3, but not CD4. On the top right-hand side, we have cells that are expressing both CD3 and CD4, and then in the bottom left-hand corner, we have those cells that are expressing neither, okay? So think of it as two axes, and each of those is an indicator of the brightness, the extent to which a cell retains the antibody and indicates to what extent that cell expresses the antigen that antibody is specific for. And so if we then compare an individual that's uninfected or is HIV infected but does not have AIDS, what we would see is that there's a healthy population of CD4 T cells in the top right-hand quadrant. So we're looking at the left-hand side now. Whereas an individual that has progressed to AIDS, we can see that although the bottom two quadrants are very similar, the top quadrant, the CD4 T cells, is largely diminished. And if we were to calculate that works out to less than 200 cells per millimeter cubed of blood, that individual would be classified as having acquired immunodeficiency syndrome. So in terms of diagnostics, AIDS is just one aspect or one disease which can be diagnosed using flow cytometry. PNH is another disease, and this is characterized by red blood cell lysis as a result of the absence of CD55 and 59 molecules on the surface of those red blood cells. And so we can use flow cytometry to interrogate the expression of CD5 and 59 on the surface of those cells in order to diagnose this disease. Acute myeloid leukemia. The co-expression of two molecules entitled CD13 and CD34 on blood immune cells indicates enhanced production of immature immune cells, right? And in terms of research, the applications of flow cytometry are endless. They're used in every single lab almost every single day. In terms of vaccine efficacy, though, I'll just highlight this one example. If you were testing a vaccine in an animal model, you could then look for vaccine-specific T cells and B cells within that animal after a certain amount of time. So it's a really interesting way to interrogate cellular populations. So kind of in summary, if you want to identify soluble molecules to perform a diagnosis, such as you're looking for rheumatoid factor in the serum of patients that you suspect may have rheumatoid arthritis, or if you want to detect HCG in the urine to detect pregnancy, ELISAs are your go-to. If you want to detect cells of interest because you want to diagnose AIDS, for instance, you want to look for absence of CD4 T cells, flow cytometry is the way to go. Either way, antibodies are absolutely integral to this process. And antibodies used in this fashion have changed the world, right? Even in 1984, so a long time ago, the few individuals that were responsible for the discovery of the principle for production of monoclonal antibodies were awarded the Nobel Prize, right? Because they've been used in so many facets of research, diagnostics, as well as Danya will talk about how they can be used to treat individuals with various diseases. So antibodies really have changed the world. And with that, I'd just like to say thank you and ask if you have any questions.